with cancer and is in remission, or know somebody with currently cancer, you're going to want to hear this presentation. Okay, we'll get started. When you get diagnosed with cancer, it's a very personal and unique situation. Understanding actually what your tumor is doing before it's treated is critical to getting the best treatment, the less toxic treatment, and the most cost-effective treatment around. Helomics is a personalized medicine company in oncology. We do comprehensive tumor profiling. What we look at is live and fixed cells. So when a patient's diagnosed with cancer, the tumor cell is removed. It's sent to our laboratories in Pittsburgh, Pennsylvania. We go through a proprietary set of analysis while we'll grow the patient's cell outside the body. At the end of the growing period, through all the analysis, we then test the patient's tumors with the drugs that they will eventually be tested with, and then tell the doctor before the patient's been treated, virtually, which drugs will work, which drugs won't work, and which drugs will work for a period of time. It's truly personalized medicine. We're the only company currently in the world that does this around comprehensive tumor profiling. We have over 150 employees and tens of millions in revenue. We're backed by private equity. We have a very experienced management team that's been doing this for over 30 years, all in the leading areas around personalized healthcare and oncology. We've built a company by starting with the end in mind. We're a reverse startup. So private equity backed, we're buying companies with technologies and reorientating technologies to do one very important thing: comprehensive tumor profiling on a very unique and personalized basis, but more importantly, on live tumor cells. Because as soon as you preserve the tumor cells, they start to change the DNA protein. They're not representative of the original patient tumor that's growing in the body. We're also doing world-class partnerships. The 100,000 Cancer Genome Program in England, we're working very closely, as you can see, with the top pharmaceutical companies to elucidate the differences between cancers amongst all of us around the world. So why do this? Because one drug doesn't fit all. This is the mantra that we've had in the pharmaceutical industry for decades. We know now that not 50% respond and don't respond, but there's degradations and elucidation that occurs when you do this. So it's the right patient, the right dose, the right time, at the right cost. Cost affordability is a big part of our healthcare problem today, but more importantly is finding the right medicine the first time. Because sometimes you can make the cancer worse by giving a drug that beats the immune system down. And then other drugs can't take over, then they switch to trial and error, and by then the cancer is spread. So what we're trying to do here is not just tell you which drug to pick, but is it the normal dose? Is it the lower dose? Is it the higher dose? Is it combinations or single? Or should you forget about that drug and look at other drugs? This is truly the notion around personalized healthcare and oncology. Why? Because the body's very complex. Tumor biology is very complex. I'll ask you, the doctor, which of the cancer cells, because they're all cancerous, are the ones that are going to metastasize and spread your disease? Are they the blue? Are they the red? Or are they the mixed cells? So we're not as personal as we think we are in tumor biology or treating on a personalized basis. Now, let me give you an example. I'm flying over in an airplane and I'm looking at rivers, valleys, green topped houses, blue topped houses, and red roofs. How do I know which of the houses and bridges and rivers that are ones that are going to cause the cancer to metastasize versus the ones that don't? Understanding the tumor biology, not just of the whole tumor, but bits and pieces down to the cell, is the critical part in a live cell analysis. You capture everything that's living within the patient. For example, this is a blood cell. If a lot of these cells here are involved in angiogenesis and you have a lot of blood cells, that is not a good thing. There are certain drugs you do not want to be given. Only comprehensive tumor profiling on live cells can actually give you this information. So what we look at is DNA, the sequence and all the variations when it turns on, the protein sequence and the proteins turning on. The most important graphic on this slide is the blue box. It's the cells in your body that are multiplying when they're cancerous and understanding the difference when they're multiplying. This is the cancer cells going through the full cell cycle. Some cancer cells go in about 7 hours, some 36 hours. The question is, why is cancer different in, say, two patients with ovarian, the same age and the same ethnicity? Why? Until we understand the underlying principles of individual live cells on a comprehensive basis, we'll never really understand what's causing that tumor to behave the way it is. Hence, it's going to be difficult to treat it. Now, what we do is we take fixed cells, these are cells that are put in formalin and other preservation methods, and we compare them with live cell analysis from the patient tumors. We go in the research market, the clinical research market, and yes, we're selling products and getting reimbursed by commercial payers, including Medicare. So we're on market. If you get diagnosed with cancer, the first thing you should be asking for is finding the right oncologist and getting access to some of the products and services that we have to truly characterize your tumor before the first line of treatment occurs. What we offer is a comprehensive analysis on the specimen itself, the pathology that I just shared with you, the topography analogy with the plane, genomics, proteomics, and we overlay all of that, and then we look at the tumor biology that's still growing, and then that makes a full profile. And then we can say, well, now when we treat you with a particular drug, I'll just switch over here. So, as we treat you with a particular drug, okay, I'll just go like this. We'll be able to tell you the degree of which that drug will work or won't work, and also tell you the combinations. Can you hear me? A miracle. So then when we get to this point, when we tell the physician which drugs are working and which drugs aren't, we can go back and say, well, the cells look like this, the gene sequence is this, the structural variation is this, here's how the proteins work, here's how the translation is at the top of the cell, and oh, by the way, there's a lot of fibrosis in that tumor, which means there's a lot of scar tissue. That's why some of the drugs aren't getting to the cancer cells to kill them. We have a picture worth 1,000 words, but it's also worth 1,000 words of data that helps elucidate specifically the way your doctor wants to treat the patient on a personalized basis. So we're still not there yet. We're up here at personalized. This is getting it down to where I can actually pull together graphically and data-wise how this tumor is progressing. Simple process. We collect the specimens from the cancer patient depending on what stage they are. I'll just finish with this. Must be my magnetic personality. Also, I'll just finish up with this. And then we look at live cell and fixed cell, because a lot of times these are put in biobanks at cancer centers, so we want to compare what they have from past samples with current samples. We try to get the very first one before you've ever been treated, or neoadjuvant therapy. And then during courses of treatment, your cells are constantly changing, so we want to look at the profile of the cells over the life of your cancer. And then when we get to the end, there's 3 different drug elements that you could go for here. It is small molecule, biopharmaceutical, which is IV, the chemotherapy of today. And we're on the cusp in the near future where we're looking at immunotherapy. So we'll take your cells out of your body, we'll put the right payload in and put the body back in motion by putting the cells back in, and the cell structure will then make the new healthy cells. And that's immunotherapy. That is one of the forefronts right now with 5 pharmaceutical companies currently in clinical studies for cancer. So one of the tests that we offer, our flagship product that's reimbursed, is called ChemoFX, and this helps understand where chemotherapy comes in, which is the first line of therapy right now for most of the drug classes, although we're getting drugs that are less toxic outside of the chemotherapy, and there's new generations of chemotherapy drugs as well. We've got 10-year clinical study, lots of data to show it's cost effective, as well as clinical medical utilities. When you use this test, on average, quality of life goes up, the overall survival increases by 17 months, and the progression-free survival when the cancer is put in check before it comes back is over 50%. Do you know how many birthdays, how many anniversaries, how many graduations, and how many holidays that is? If this was a drug, we'd be a blockbuster company. So just think about this for a second, where we are with our current line of therapy. Our clinical focus of products currently, and we'll be expanding this pretty rapidly over the summer into the fall. Our CellFX is our platform for analyzing cells on a research basis. We work closely with pharmaceutical, big biotech, and major cancer research centers around the world. So in summary, Tumor biology is very complex. You have to understand the full complete tumor cell by cell. Comprehensive tumor profile from a biologic standpoint is how you do this, not just genes, the genes, proteins, and the whole complement. Otherwise, we don't know what we don't know. We offer the full complement of this, and we're looking for collaborations at this meeting and partnerships. We're also looking to buy and license technology. A very streamlined process because we work very closely with the physician and the caregiver team, the cancer center, during the course of treatment. It's a very personalized approach. Once again, our clinical focus, based upon the true cell proliferation of everyone's cancer itself, personalized and unique. So in the end, we can tell you which drug to give, what dose, how often, and how effective it's going to be before the first administration of the drug goes into the human body. So we're not wasting toxicity, we're not beating up patients, and we're not wasting money in our healthcare system. This is what we're all about. We're based in Pittsburgh, Pennsylvania. I want to thank you for attending the presentation. Our booth is right over here, 109, within the Pennsylvania Pavilion. And please remember, if you know anybody with cancer, diagnosed with cancer, or someday that happens, please remember Helomics.com. Thank you very much.